All right, so this is going to be our lecture on cells and the fact that cells are our most basic unit of life. So you can see in the background of this intro slide, you can see varying cells. You can see cells as they begin to divide in a process known as mitosis to create new genetically identical cells. And so we'll go through, we'll talk about different cell features and functions. We'll talk about different characteristics of cells as it relates to um, protein synthesis, different types of transport into and out of cells, and various other things like that. So we'll get started here talking about kind of the origin of cells and rather who discovered them. Okay, so Robert Hooke in the mid 1600s, 1665, examined cork tissue under a microscope, and what he saw was this network of these little chambers that he called cells. They resembled jail cells to him, in this idea that they are all divided from one another. And then about 10 years later, in 1675, Anton van Leeuwenhoek was actually the first person to discover and see living cells underneath a microscope. So this changed lots of biology leading into the 1700s and the 18th century because of these amazing discoveries, and they also led to lots of new information in the field of microbiology. So this is what Robert Hooke saw. This is a picture of dead cork tissue, and then these were his drawings. So you can see this is what he was referring to as these cells, these little blocks that are divided and separate from one another, but really close and really adjacent to each other. So this is what he saw. This is what he ended up drawing to represent what it was he was looking at. And so we'll take this basic drawing from Robert Hooke and we'll expand on it, and we'll talk about all different types of features and characteristics and functions of different types of cells. So in the late 1700s, so the 18th century, there was a theory proposed known as the cell theory. And there are three main parts, and there are renditions to the cell theory that include a fourth part, and some include more parts than that. But these are what we will focus on. So the fact that the cell is the most basic unit of life. We know this from our hierarchy of organization. You start with small non-living atoms and molecules and work your way up to organelles, and then finally cells being the most basic unit of all living things. We know that all cells will come from other cells via cell reproduction or mitosis. So there is specific stages of mitosis that we discussed, talking about prophase, metaphase, anaphase, telophase, and what's happening at each of these to produce two new genetically identical cells. We know that all living things are made of cells. Okay, so we know that cell is the most basic unit of living things, and all of these living things are then composed of cells. And then this fourth one is one that is sometimes added and may not be a part of all cell theories that you read, but biochemical activities of cells are controlled by subcellular structure. So what this is talking about is our organelles. So we have organelles that are subcellular, they're beneath the cell and within it, that all have a very specific function. We'll take a look at those functions later in the lecture. But it's these organelles that are producing what is happening in the cell, and then we know when lots of cells begin to function together, we get tissues, and you can move up to organs, organ systems, and ultimately an organism. So when we look at a cell, and this is a picture of a eukaryotic cell, we have eukaryotic cells. We have a plasma membrane, and we'll take a look at the plasma membrane. We have a cytosol, and the cytosol is referring specifically to the fluid part within the cell. And then there's something called cytoplasm, and cytoplasm is the cytosol, so that fluid part, plus all the organelles. Okay, so cytosol itself is the water fluid part of the cell. If you take that part, add organelles, that's your cytoplasm. Okay, sometimes you'll see them used interchangeably, and for the most part, that's fine, kind of referring to everything outside of the nucleus. But there is a technical difference between cytosol and cytoplasm. And then nucleus, this is the largest of our organelles. It is generally located in the center of cells. On histological stains and tissues, you can see the nucleus generally pretty well, and sometimes the nucleus gives you a clue as to what specific cell type you're looking at. For instance, when you're looking at different types of leukocytes or white blood cells, looking at the nuclei or the nucleus of the cell will give you a very good indication as to what that cell type may be. So this is sometimes referred to as the fluid mosaic model of the plasma membrane. And what that's meaning is fluid. So if you think of if something is fluid or there's fluidity, it's able to change, it's able to sway and adapt to things around it. And that's exactly what this plasma membrane is able to do. It's able to twist and turn and accommodate different changes that the cell must undergo. It's also mosaic because there are lots of different pieces. So if you think of the term mosaic in terms of art, there are different pieces and shapes and colors and sizes to art. Well, that's kind of what you see here. We see all different types of glycoproteins and glycolipids. You see proteins that span the plasma membrane. You see some that exist only on the surface. You see these little yellow structures here. This is cholesterol. And so collectively, we call this whole thing the phospholipid bilayer. And we'll take a look a little more closely at what it primarily makes up our phospholipid bilayer. So here's another picture showing nucleus, cytoplasm, as well as our plasma membrane around the edge. So cell shapes, this is generally a heavy lab topic, but we'll talk briefly about cell shapes. So we have squamous cells that are flat, stellate cells that are more star-like, and then cuboidal cells that are cube. We have columnar cells, cells that are spindle-shaped or fusiform, and then fibrous cells like skeletal muscle cells. And here's a picture of all of those. So flat, star-like, cuboidal, columnar, fusiform are very kind of spindle-shaped, and then fibrous with our skeletal muscle cells. So our plasma membrane, this is the main feature that we see if you're talking about kind of the external aspect of a cell. It's what you see before you get to the internal components. And so it is what you can call selectively permeable, meaning that it controls selectively what goes into and out of the cell. So into it from the environment and then out of the cell going into the environment. And the primary component of our plasma membrane is our phospholipid. Okay, it's called a phospholipid bilayer because it consists of two layers. There is one on top, oriented with their fatty acid tails hanging down, and there is a layer on bottom with their fatty acid tails oriented upward. Each of the heads and tails can either be classified as hydrophobic or hydrophilic. The heads are hydrophilic and tails are hydrophobic. And what hydrophilic and hydrophobic mean is water fearing or water loving. So hydrophilic is philia, refers to love or attraction. So things that are hydrophilic are attracted to water. And then likewise, things that are hydrophobic, phobia meaning fear, they fear water or do not have an attraction to water. So if you were to drop phospholipids into a tub of water or something like that, they would orient themselves in this fashion because of these characteristics of their heads and tails. So here's the same thing, just in a slightly different view. And then here's a picture of an actual phospholipid. You can see a phosphate head with kind of this glycerol neck region, and then you have fatty acid tails coming down. Note that one is saturated, one is unsaturated, so you get that double covalent bond between two carbons, and you get that little kink in this fatty acid tail. Now, when we look at our plasma membrane, we don't just have phospholipids, but we also have proteins embedded within it. So you can classify the proteins broadly in one of two ways based on whether it crosses the whole plasma membrane or if it is only on one surface. So if it crosses the whole plasma membrane and it's a large part of it, it is termed an integral protein, meaning that it is integrally a part of and embedded within this plasma membrane. And then we have the opposite of that, where something is just located on one side, either closer to the inside or closer to the outside, and that's going to be a peripheral protein. Okay, we'll see that some of these act as receptors. Some of these will act as channels to allow different ions and solutes to enter and exit the cell. So when you see here, we see a peripheral protein located on the periphery of the plasma membrane, and here's an integral protein that is spanning the entire width of our phospholipid bilayer. Now, we said that these proteins can have different functions. I said one is a receptor and one is a carrier, but you have other functions as well. So you have things like cell identity proteins that tell our immune system that that particular cell is self. It is a normal body cell. It is not foreign. You have receptor proteins that bind to different things called different ligands, which are different molecules that will result in a signaling cascade. You have enzymes, and you have motor proteins. And we'll talk about what all these different types are as we progress. So a channel protein or a carrier protein, these are two different types that allow solutes to be able to cross our phospholipid bilayer, but they allow things to cross in a slightly different manner. A protein channel, as you can see, is going to allow things to flow freely across, generally from an area of high concentration to low concentration. So things like gap junctions. These can be things like this that allow things to cross. And then we have a carrier protein that's actually going to bind a solute and then undergo a conformational change in its protein structure to put that solute on the opposite side. So it goes from outside the cell up here to inside the cell down here. We also have receptor proteins. So this is a process called a G protein or a G protein coupled receptor. And so what this is talking about is this idea we have a receptor protein right here, this adrenergic receptor that will bind maybe a catecholamine like epinephrine, and it is closely associated with something called a G protein. And so these ligands, this epinephrine, it's not able to diffuse across this membrane and enter the cell's internal environment. So what it has to do is bind to a receptor. That receptor activates a G protein. This G protein converts something known as guanine triphosphate, to guanine diphosphate. And then an enzyme known as adenylate cyclase will convert the it will convert ATP to cyclic AMP. And then the cyclic AMP is what we'll call a second messenger. And so that second messenger can then go within the cell to perform whatever function it needs to for this particular ligand. So these molecules that bind to this G protein coupled receptor activate a second messenger within the cell to exert its effect. Whereas you can have some things that are lipid soluble, so things, certain hormones are lipid soluble, that are able to directly cross this plasma membrane, enter the cell, and exert its action within the cell directly, as opposed to acting on one of these second messenger G protein coupled receptor mechanisms. So we can also have proteins acting as enzymes. So what we can have is an enzyme that acts on a particular substrate, and a substrate is just meaning some product or some molecule that this enzyme is going to bind and do something to, and then it can result in it changing form. So maybe you take these two examples here, you're going to take this red piece and this yellow piece and join them together, and then you make this one orange piece. But note that the enzyme remains unchanged. So what this means is that this particular enzyme is capable of redoing this process over and over again, and it's not just a one and done type of process. You can also have proteins act as catalysts that serve to lower the activation energy required for chemical reactions. And so this E subscript A is referring to the activation energy that must be overcome in order for a chemical reaction to proceed. Everything from this dotted line down, this delta G, is referring to the change in energy. This is actually what is happening, but you have to overcome this activation energy in order for this to happen. So you can imagine this is going to take some time. Now, if you have a enzyme that's acting as a catalyst, what it does is it can lower this activation energy, And so you can imagine if we go from this upper activation energy to this lower one in red, this reaction is going to proceed much quicker. Now, you get the same delta G, you get the same uh, change in free energy, but the time at which it takes is much faster when you have a catalyst involved. Now, we will answer a few questions and we'll go on. So phospholipid heads are, you should think that phospholipid heads are hydrophilic and phospholipid tails are hydrophobic. We know that the cytoplasm contains two main components. That's going to be our cytosol as well as the organelles. And we know that if a protein is embedded within the plasma membrane, if it's completely embedded within and crosses the whole surface, it is going to be an integral protein. So we will stop there and we will pick up with part 2 in the next